The sexes can differ in terms of how much they invest into reproduction, and this is often used to explain the sexual dimorphism that is common in many polygynous species. Females, who have high reproductive costs, benefit from choosing high-quality males, resulting in greater intensity of sexual selection on male ornaments. However, males of many highly ornamented species appear to be both competitive and choosy. But male mate choice is usually attributed to traits that indicate fecundity, which can be uniform across females of many species. This means that evidence of male mate choice does not necessarily imply that male choice contributes to behavioral isolation among species, and in most cases, behavioral isolation is thought to be primarily driven by female preferences for conspecific male ornaments. In the present study, we sought to determine whether male preference for conspecific female visual signals contributes to behavioral isolation between two sexually dimorphic darter species, Ethiostoma barronense and Ethiostoma zonale. Males in breeding coloration are pictured on the top row, and females of each species are shown below. To measure male preferences, we use a dichotomous choice assay consisting of a central test tank positioned between two stimulus tanks. A focal male was placed in the test tank, and a conspecific and heterospecific female were placed individually into each stimulus tank. Stimulus females were size matched to help ensure that male preferences, if observed, were based on visual signals unrelated to fecundity. We marked the test tank with two 5-centimeter preference zones, shown here in red, on each side closest to the stimulus tanks. Over the course of a 15-minute trial, we recorded the amount of time the focal male spent in the conspecific and heterospecific preference zones. Time spent in either preference zone relative to the entire trial time was used to assess male preference. We also calculated male strength of preference using the equation Tc minus Th over Tc plus Th, where Tc is the time spent in the conspecific preference zone and Th is the time spent in the heterospecific preference zone. This score ranges from +1 to -1, indicating a complete preference for conspecific or heterospecific individuals respectively. Comparing the proportion of time in each preference zone, we found that male E. barronense, shown on Figure A, and male E. zonale, shown on Figure B, significantly preferred conspecific females. This suggests that male preference contributes to behavioral isolation for this species pair. We then compared our data on male preferences to previously published data on female preferences for the focal species. In both figures, males are shown in dark gray and females in light gray. Looking at Figure A, we see no significant difference between time spent in either preference zone for male and female E. barronense. Additionally, male and female strength of preference was not significantly different for this species. However, for E. zonale, shown on Figure B, we see that males spent a significantly larger proportion of time in the heterospecific zone than females. We also found that for E. zonale, male strength of preference was significantly lower than female strength of preference. Because male preferences, as well as female preferences, may contribute to behavioral isolation, we next estimated the relative contribution of each sex to total behavioral isolation. For the focal species, males initiate courtship by approaching a female. Females then either accept or reject a male, and, if accepted, other males often attempt to supplant the original male. Thus, we assume the relative order in which male and female behavior contributes to behavioral isolation to be male mate choice, followed by female mate choice, and then male-male competition. We consider these to be 3 barriers that contribute to behavioral isolation. Measures of male-male competition and female choice came from previously published data. We then calculated total reproductive isolation due to behavioral barriers and the absolute contribution of each individual barrier, weighting earlier acting barriers more heavily than later acting barriers. This is because a given reproductive barrier only eliminates gene flow that has not already been prevented in previous stages of isolation. The relative contribution of each behavior is then calculated as the ratio of its absolute contribution to total reproductive isolation. We found that total behavioral isolation between the focal species is nearly complete, with a total isolation score of 0.97. Taking the absolute contribution of male choice, female choice, and male-male competition over the total isolation score, we found that male mate choice contributes as much as female mate choice to total behavioral isolation in E. barronense and E. zonale. In summary, our results show that males of 2 sexually dimorphic species of darters prefer conspecific females, and that strength of preference for conspecific mates is just as strong for males as it is for females in E. barronense. Combining our results on male preferences with data on female preferences and male-male competition, we found that male mate choice contributes as much as female mate choice to total behavioral isolation. This indicates that males likely play an important role in behavioral isolation for these sexually dimorphic polygynous species.